Hello, the purpose of this video is to go over the allele frequency data analysis lab for chapter 17, section 2. So the information for this is in your e-text or in your textbook on page 490. So this is basically what we're doing here on page 490 is we're analyzing this data. And the information that helps you be able to do the formulas is right here, the Hardy Weinberg principle. So what I'm going to do is take you through this worksheet and give you some hints on how you can use the formulas that are down here at the bottom to determine allele frequency. So what I've done is I've taken the relevant information and put it on a PowerPoint slide so that I can just stay on this one screen. All right, so what we're going to look at is allele frequency. And there's something called the Hardy Weinberg principle, which we can use to predict frequencies of certain genotypes if you know the frequency of other genotypes. So let's say you have a genetic condition that's controlled by two alleles. So we have a big S and a little s. So our big S, let me get a black pen. A big S is going to be our dominant trait. Okay, so that's our dominant trait. Our little s is going to be our recessive trait. Okay, now the condition affects only homozygous recessive individuals. So we have three different genotypes possible. We have big S, which is homozygous dominant. We have heterozygous, which has one of each. Remember, hetero means different, big S, little s. And then we have little s, little s. Now, the only genotype that would exhibit the disease or the condition, if it's not a disease, would be the little s, little s. If you have big S, you don't have the gene which can cause the trait. Big S, little s does not show the trait because the big S is dominant, but they can pass that trait on to their offspring. So what we're wanting to do is to calculate the allele frequencies. So we're going to use this Hardy Weinberg principle and use these formulas here. All right, so when we use these formulas, they're using P and Q. And just keep in mind that P and Q represent the dominant and the recessive traits. So in this case, the big S is our dominant, so that's going to be equal to P. And then our recessive is going to be equal to Q. So let's see how we can figure these out. All right, they give us a population. They tell us that we have 10,000 organisms in our population, and only 36 have the condition. All right, here's where I'm going to go Dora the Explorer on you. I'm going to ask a question and pause, and I want you to think of Dora looking at you. I don't know about you, but when Dora looked at me, I thought she was staring into my soul, and I felt obligated to come up with an answer. So I'm going to pause for you to reflect, and when it comes to doing the calculations, I'm not going to do the answer. I'm going to show you how to get to the answer, then have you calculate it. All right, so we know that we have 36 individuals out of 10,000 that have the condition. So which one of these genotypes are those 36? I'm doing my Dora pause. All right, so you know that the condition is only exhibited if they're homozygous recessive. So that would be little s, little s. So we know that 36 of these organisms have the little s, little s. Now, the Hardy-Weinberg principle only works when you're in a population that's not changing. So that means there's not any kind of situation that's causing some of these alleles to get selected out. A good example, let's say you have snowshoe hares, which are white. But let's say that if you have little s, it makes your hair color black. So you would stand out in a white field and you would probably become lunch for someone. So that would probably select out the little s's, so you probably wouldn't have any black snowshoe hares. That probably may have happened to that population at some point. So we're assuming nothing like that is going on, so we can use these formulas. So they give us Q squared is equal to 0.0036. So now I'm going to go mathematical on you. So if we want to turn this into Q, we have to do whatever the opposite of squaring is, and that's square root. So instead of my handy dandy Blues Clues notebook, I'm going to pull up my handy dandy calculator, and I'm going to take 0.0036 and I'm going to apply the square root. Not the square root. Yeah, the square root. All right, so what did we just calculate? If I took q squared and I did the square root of that, what did I just come up with? Yes, I just came up with Q. So Q is equal to what was it again? Let's go back here. 0.06. So to convert that to percentage, you move the decimal twice, so that is 6%. Sorry for my terrible writing. So you just figured out, question 1, what are the frequencies of the big S and the little s? So we just figured out the little s. Now there's a formula here that says P plus Q equals 1. So if we have S, which is Q, equal to 0.06, what must P be? All right, so calculate that out, and that is the answer for big S, which is your dominant trait, which is represented by P. So the frequencies, S is equal to something, little s is equal to 0.06. They both add up to be 1. So that's how you can figure out the distributions of the big S and little s. All right, so the next question is we're going to calculate the frequencies of each of these genotypes. So that formula is here. So this is how we can do this. We can figure out P squared by taking whatever you came up for big S, because remember, big S is P and Q is little s. So you know what big S is now, so you can figure out P squared, taking your P, squaring it, and you now have your answer. 2PQ is you take whatever you got for P, whatever you got by Q, multiply those each other, then multiply by 2, and that's going to be your big S, little s genotype. And then finally, we already had Q squared. They gave it to us. So that's your distribution for little q, which we figured out up here. So little s is this. So they gave you that answer. All right, next you're going to calculate what percentage of people in total is likely to be carrying the S allele, whether or not they know it. Now, if they have little s, they know they have it because they're exhibiting the phenotype. They're exhibiting the trait. What other genotype could possibly carry the S allele, but they may not know that they have it? You are right, big S, little s. So to answer this question, to calculate the percentage, you would take whatever you got for big S, and whatever you got for little s, and add that together, and that would be your answer. So there you have it. That's how you can complete this lab. Hope this helps. And if not, send me a message and I will answer it as soon as I can.